This video is going to cover the circulatory system. We start out in this chapter by just mentioning, what's the point of the whole system? So why is blood so important for life? It's because it transports everything that your body needs. It transports your respiratory gases, oxygen and carbon dioxide. It transports your nutrients like glucose and amino acids. It transports waste so you can remove them. That would include ammonia and urea. It transports hormones around your body from the endocrine system. It's transporting everything. And so that is why blood is so important for life. It transports literally everything that your body needs. What's the function of the heart is the next thing that you have. It is the pump of the circulatory system. So it's what drives the transport of blood all the way around in your body. So it acts as a pump to move the blood. Compare and contrast open and closed circulatory systems. Open circulatory systems, the blood vessels are open-ended. What that means is the blood does not stay in the blood vessels at all times. It leaves and it mixes with the interstitial fluid and it becomes something that's usually known as hemolymph. So open circulatory systems are, again, open blood vessels. Blood does not stay in them. It's not a very efficient system, but it's good for some organisms, like this grasshopper happens to have an open circulatory system. Closed circulatory system is what we have. The blood stays in the blood vessels, barring some type of injury. It's a more efficient system. It tends to be better at transporting materials because we're able to keep pressure higher, and so it allows for more activity and just better organ function as a whole. So again, for closed, the blood stays inside the blood vessels. The blood vessels form a continuous loop from the heart and then back to the heart. I do also want you to mention organisms that have each of those. So for an open circulatory system, an example of that would be any arthropod. That's A-R-T-H-R-O-P-O-D. A grasshopper is an example of an arthropod, but so is any insect or spider or things like that. A closed circulatory system, vertebrates have that. That includes fish all the way up through us, humans. Next, how many chambers does a fish heart have, and what are the names of the two chambers? So fish have a two-chambered heart, unlike us. The names of the two chambers are up here on the slide. They have one atrium, which receives blood back from the body, and then they have one ventricle that's going to discharge or be the actual pump that pumps blood back into circulation. So two chambers, one atrium, one ventricle. How many chambers does a frog or reptile heart have, and what are the two circulations that are starting to develop? Frogs and most reptiles like snakes and lizards, they're going to have a three-chambered heart. They have two atria and one ventricle, like you see in this picture up here. What are the two circulations that are starting to develop? So on the right side of the heart, we're developing a pulmonary circuit. In the case of frogs, it's a pulmocutaneous circuit because a lot of their respiratory system is actually in their skin and not in their lungs, and so that's why it has a different name from what ours is going to have. The left side of the heart is called the systemic circuit. Fish don't have that. They just have one circuit. The heart has to pump blood to the entire body, to their respiratory system, which is their gills, and then to the tissues, all with that same ventricle. Here, we're starting to become a little bit more efficient, but we still have this problem where oxygenated blood can mix with deoxygenated blood in the ventricle, which means the blood that we pump out into the systemic circuit is not as oxygen rich as it could be if we went ahead and divided this into a four-chambered heart. How many chambers do birds, mammals, and also crocodilians, the crocodiles, alligators, caimans, things like that, they also have a four-chambered heart. What are the names of the two chambers? We get two atria, a right and a left, and two ventricles, a right and a left. What are the 7 functions for blood listed in table 29.1? This is the 7 functions that you see up here. You can pause the video so that you make sure you get that. I mentioned most of those things as I was talking about just the importance of blood, so just pause your video to get what you need. What is plasma and what can be found in the plasma, and about how much blood is made of plasma? So that's a lot of questions for one thing. What is plasma? It's the extracellular matrix found in blood. What can be found in plasma? It is mostly water, but there are a bunch of dissolved solutes within the plasma. So if you don't remember what a solute is from Biology 1, that's material that gets dissolved in water. And in the case of plasma, that includes everything blood is trying to transport, respiratory gases, nutrients, waste, hormones. It includes vital proteins from your immune system that are called antibodies. It includes clotting factors like calcium, thrombin, and fibrinogen. So your plasma, while it is mostly water, there's a bunch of material dissolved in that water that happens to be critically important for your function, including the stuff that it's supposed to be transporting. About how much of blood is made of plasma, it does vary dramatically from one person to the next, but on average, it ends up making up just over half of the blood, so about 55% of total blood volume. Next, what is the function of red blood cells, and what protein molecule do they contain to help them do this? Red blood cells really just have one job, and that's transport oxygen. Everything about them has been designed to really do that one thing and do it really well. What protein molecule do they contain to help them do this? It's called hemoglobin. That's Hemoglobin. Hemoglobin is a genetically determined protein with a special little molecule called heme inside of it that contains iron. The oxygen binds to the iron so that you can transport oxygen around in your body. Now in this picture, all of these little pink cells, they're all red blood cells. And what I want you to notice is they don't even have a nucleus. Like I said, they have been designed to specifically carry oxygen, and if they had a nucleus, that would be a bunch of space taken up by DNA, which doesn't transport oxygen. So they actually kicked the nucleus out so they can transport oxygen that much better. Okay, let's talk about blood typing is the next thing that you guys have in your notes. So there's 4 major types of blood that are dictated by what's known as the ABO groupings. We talked about this a little bit in genetics last semester, but what it amounts to is people have antigens on the surface of their blood cells, and those antigens are what determine what blood type a person is going to be. So, for example, I'm going to start up here with our person who has type AB blood. On their blood cells, they have 2 differently shaped antigens. There's an A antigen and there's a B antigen, and because they have both of those, they have type AB blood. A person who has type A blood, they just get one of those antigen shapes. They don't get the other one, so that's why they're just type A. Person who is type B, they just get the B shape. They don't get the A. And a person who's type O, they don't get any antigens. That's why they're called type O. It's because they're actually like sub-zero. They just don't get any antigens. Now, what complicates this blood typing is that we also have antibodies that are circulating in the blood, and antibodies are designed to fight other blood types that you might get exposed to so that that other blood does not hurt you. And everybody has the types that correspond to opposites, essentially. But here's the thing. Type AB blood, they actually don't get any antibodies because that would mean that they had to attack their own blood. Oh, darn, I ran out of room. Antibodies. And so AB blood doesn't get any of those. Type A blood, they get what's known as an anti-B antibody. So antibodies are these Y-shaped proteins, and they have shapes at the top that correspond to the antigen that they're trying to catch. So this shape corresponds with this shape. So that means type A blood has antibodies that can come, nope, you go away, window, that can come down here and could try to murder this blood type if type B blood got exposed to type A blood. Type B blood has the exact opposite situation. They have anti-A antibodies. So if a person with type B blood were given any A antigens, including type AB blood, that means that they would try to murder that blood. Type O is even more fishy. It has antibodies against both A and B blood. Pew, pew. Okay. Those are my little roundy shapes, by the way. So they've got antibodies against all. Now, this sets the stage for a couple of different issues. Blood transfusions are used routinely in medicine to try to help people who have suffered severe blood loss. The antibodies in a person are what do the damage. So here's the thing. Since a person who has type A blood don't have any antibodies, that means that they are what's known as the universal recipient. They can receive blood from anybody because they don't have an army. They can't fight anybody's blood, so you can give them anything you want to give them. Type O, on the other hand, it doesn't have any antigens to attack, and that means antibodies are ineffective against type O blood. That means they are the universal donor. you can give type O blood to anybody. And that's because there's nothing to be attacked on them. So that means a person who has type A blood, if they were in a car accident and needed some blood, you could either give them type A blood or you could give them type O blood. Same for type B. You could give them type B or type O. Type AB, you can give them A or B or AB or O. You can give them anything that you need to. Type O, though, can only get type O because they've got antibodies against every other type of blood. Now, advantageously, in the United States, type O blood is actually the most common blood type that's out there. However, it's always being requested by blood banks because it can be used in anybody. So we've got a lot of people who can donate it, but we also have a lot of use for it since it can be used in anybody who needs to have a blood type. Now, to complicate this factor, there is another antigen that can be present on the surface of some of our cells. There's actually several, but one of them is called the D antigen. And if the D antigen is present, then we give them a positive. And if it's not, we give them a negative. So I'm going to attempt to make a different shape, and I'm going to do it on this one. And I'm going to do it with a different color just to sort of prove a point. I'm trying to think of a different shape I can use. Okay, I guess I'm going to do a box. No, go away. Stop being weird, clicker. Okay, so if that antigen is present, then they are positive. So this person is now not just type AB, they're AB positive. Since this one doesn't get the square, they would be type A negative. If I gave them a square, they're B positive. If I didn't give them a square, they're negative. There are also antigens that can be made against the D factor, which is called the RH factor. It's different in a couple of different ways. So since this person is A negative, it means that they have the potential to make antibodies against the RH factor, but they will only make it if they get exposed. So if that person never gets exposed to positive blood, they'll never make that antibody. But if they do get some O positive blood at some point because they needed some blood, that means they're going to start making that antibody, and in the future they can attack positive blood. It's just not automatically made. All the things that are in red, they're automatically made from birth. You're making those antibodies and antigens. But on the antibody side, you only make the blue one if you get exposed to it. If you're never exposed, you never make that one. Let's see, I'll also do on this slide the term agglutination because you can actually see it. So the way that we test for blood typing is we take a person's blood and then we drop antibodies on it. And then if they have the antigens present on their blood, we clump the cells together. And that's what agglutination is. So agglutination is the clumping of cells due to antibodies. So if they clumped when we dropped anti-A on, it means they have the A antigen. If they clump when we put the anti-B on, it means they have the B antigen. So if they clumped or agglutinated on both sides, that's how we recognize type A blood. This person only clumped on the A side, which means they only have the A antigen, so they're type A. This one agglutinated only on the B side, so they're type B. And this one didn't agglutinate even though we dropped all the antibodies on it, which means they have no antigens, and so that's type O. So agglutination is a twofold thing. One, we use it to do blood typing so we can tell what blood type a person has. But two, it also shows you what happens if you give a person the wrong blood type. So if you were to give a person who has type B blood, type AB blood, what's going to happen is their antibodies are going to attack those type A antigens and cause agglutination, which clumps the cells together, and now they can't travel through the blood vessels like you need them to be able to. And so this causes massive clotting that can shut the kidneys down and then end up killing a person. That's why you don't want to give a person the wrong blood type. All right. What's the function of white blood cells? Immunity. One word answer there for you. What is leukemia? It's cancer of the white blood cells. So what happens is a person makes way too many white blood cells. And then because we make so many, we have a quality control issue where we made them, but they don't work like they're supposed to. So one of the more common symptoms that develops in leukemia is frequent infections that don't go away. And it's because even though they have a bazillion white blood cells, they don't work like they're supposed to. If untreated, leukemia is fatal every time. But if a person gets medical treatment, we've really come a long way in treating leukemia, so we can put a person into remission a lot better now than we used to be able to. What do platelets do? Platelets assist in the clotting process. They create something known as a platelet plug. Whenever you're injured, certain chemicals get released at the site of injury, and those chemicals activate platelets that normally float around in an inactive state. Once a platelet gets activated, it activates more, which activates even more. We talked a little bit about homeostasis in the last chapter. This is a positive feedback response. So platelets are activated through positive feedback to stop you from bleeding to death if you get an injury. That's where I'm going to stop this video. The next section of the notes goes away from blood and starts talking about blood vessels. So this is a pretty decent stopping point.